Kasi may klase ako. Call your classmates so we could start in a while. How many are you in class? How many are we expecting?
51 po dapat.
Let's wait a few minutes more before we start. Okay. In 5 minutes at 1:15, we will start whether we are complete or not. So please tell them to join in. This would not be a long session so that you could study for your exam tomorrow. Is it tomorrow?
Yes po, Doc.
Okay. Okay, good afternoon. Okay, so this is our review on enzymes and enzymes kinetics. So this is in no way a repeat of what you have had in your first year. So this is just a review. If you want the full lecture, you could view it again in my YouTube channel and look at my lectures for biochemistry there. Okay? So this is in a and question format review so that you will know how to attack multiple choice questions when you see them in the exam. Okay? So we start. Um, how do you remember enzymes? What would be its function and what is its role in our body? Remember
Doc, nagsispeed up po sila ng reaction.
nag? Okay. It would be catalysts of reactions. Okay. What else do you remember?
It decreases the activating energy po of a reaction.
Yes, it decreases activation energy. Okay. What else? Protein, non-protein. So these are the things that you have to remember to um, know that we are what we are dealing with. Is it a protein or non-protein?
Protein po, Doc.
So it's a protein that catalyzes rates of reactions by decreasing its energy activation. So that is your definition. Okay. Okay. So if you are asked, so if enzyme change the active site, rate of reaction will, what do you think will be the answer? Will it decrease, increase, remains the same, or will fluctuate? So your active site changes. What will happen to your rate of reaction?
Decrease po ba, Doc?
Will decrease. Okay? Would everyone agree? Sana ganito mga tanong sa seminar, no? Okay?
Sana nga, Doc.
Sana, di ba?
Thank you na agad, Doc.
Ha? Okay. Inaayos ko sana kung saan na pwede kayong mag-phone in ng sagot pero hindi ko pa naaral. Baka next year sa review. Ganun na. Pero unti-unti. Okay? So, decrease. So, you know that the active sites, so your enzyme molecules would contain special pockets or cleft called the active sites. Of course, you know that there would be changes. So, there would be enzyme-substrate complexes that would form when the enzyme binds to your enzyme-substrate complex. Okay? And there, your products leave your active site. So there are two ways that your binding site or your active sites change. So remember what are these changes? Your non-competitive and your competitive inhibition. Okay? This is again going ahead of my review, but it could not be touched when you look at active sites. Okay. When it's competitive, what happens? It could change by your another substrate would bind to your active site, thereby preventing your substrate to go to that active site. Thereby, it is decreased. So, it's decreased. Or it could be non-reversible inhibition as well. Your non-reversible inhibition binds to another site, but through its binding, changes the conformity of that binding site, thereby preventing your um, enzyme substrate binding or forming it into a complex. Also resulting to a decrease in your rate of reaction by binding to a non-competitive binding by a non-competitive or either competitive inhibition. Okay, so decreases would be the answer. Okay, so next. Okay. So you could still remember your biochemistry principles way back in first year. So I think you have answered this. The general mechanism of an enzyme is that it reduces the energy of activation. Okay. So, a review. So, mechanism of action of enzymes. So, these enzymes increase reaction rates by decreasing the activation energy. So, your enzyme substrate interactions, so formation of enzyme substrate complexes by your lock and key model and your induced fit model. So, remember, when there's an enzyme, remember your illustration wherein you are trying to make a reaction climb over a hill. Of course, if without the enzyme, your energy activation that you have to overcome is very much higher as to when there is an enzyme present, your energy activation lowers significantly such that it facilitates the reaction to proceed to make the product. Okay? So that is mechanism of action of enzymes. Okay. I prepared a slide pala. So this would be the hill. So without enzyme, lactose forming glucose plus galactose would have a high activation energy that would have to be overcome. Okay. With the enzyme, with such as lactase, your lactose forms an enzyme substrate complex and makes it easier to proceed with the action and therefore making the production of your glucose and galactose easier. Okay? So this is a more um, without graphics table on how your enzyme reactions occur by decreasing your energy activation and changes your reaction without affecting your delta G. Okay. I'm sorry, some are still trying to join. Okay. Next question. So the coenzyme is often a metal, always a protein, often a vitamin, or often an organic compound. So for you to answer this question, you have to know what a coenzyme is. What is a coenzyme? Anyone remember what is the answer to this? What would be the answer to this question? Or anyone who would want to hazard a guess?
Doc, try lang po.
Try.
Always a protein po pa siya rin, Doc.
Always a protein. Remember, your enzyme is already a protein. Testmanship. Your
Metal po.
enzyme is already a protein. So what would your coenzyme most likely be?
Metal po.
Metal? For sure, it is most likely a non-protein. Okay. So tanggal na si B.
Inorganic compound po ba, Doc?
Inorganic compound. Sige, let us review. Okay. These are the types of cofactors. So we could ask you what a coenzyme is, what is a prosthetic group. So non-protein bound, that is loosely bound to your apoenzyme by a non-covalent bond. Okay, so your protein is removed. Okay. Non-covalent bond, usually what would be a metal? What kind of bond would metals end up with? It is usually a covalent bond. Okay, so examples. Okay, so the answer for this question would be vitamins or a compound derived from vitamins. So it would, what are vitamins? These would be fat soluble or non-fat soluble substances that would be devoid of a protein component or as such. Okay, prosthetic groups, they are also non-protein. Okay, this is tightly bound to your apoenzyme. So this is how it would differ with your coenzyme by your covalent bonds, and this is usually called a prosthetic group. Okay, so you have to know the types of cofactors and how it behaves and what would be its components. Okay, this one. So another favorite question that we ask you would be asking your enzyme, and remember that it is grouped according to its function. Okay. So an enzyme that joins the ends of two strands of nucleic acids is what?
Ligase.
Ligase. So what's the confounder here? Anong nagpapagulo with this question? Joining. So, baka synthetase ang sagot. Correct? So, it may confuse you, especially if naghalo-halo na yung inaral nyo sa... Ay, nawala yung ano, sorry. Another. So, enzyme which helps in changing the shape of the molecule is called what?
Isomerase.
So, isomerase. Okay. So, for you to be able to answer these types of questions, okay, you have to be familiar with the functional classes of your enzymes. So, this would be the functional classes. So, oxidoreductase, transferase, hydrolase, lyasase, isomerases, and ligases. Okay. So, by definition, your oxidoreductase would be involved in your any oxidative reductive reaction. Okay, transferases would be responsible in the transfer of exchange of your um, enzyme substrates and your products. Hydrolase, hydrolysis, or anything would be that would be related to your hydrogen ions. Okay. Your lyasase would be removing. Isomerase would be, as mentioned in the question, change in structure but same. And your ligase would be joining. Okay, and that is through nomenclature you could be able to answer these types of questions. But again, basic knowledge. So there's not anywhere is synthetase found in your classifications. Okay, so for example, if you answered synthetase, so alam your professor or the one who gave you the exam would have known that you are just guessing. Okay. So this one. So which of the following is produced with the combination of an apoenzyme and coenzyme?
Holoenzyme.
Holoenzyme. How did you arrive with the answer? What did you eliminate first?
I know po na yung holoenzyme po is APOENZYME plus cofactor.
Okay. Your holoenzyme is your APOENZYME plus a cofactor. Of course, your enzyme-substrate complex, enzyme product complex, you know what it is composed of. It is your enzyme plus your substrate, your enzyme plus your product. And for the previous slide, we have reviewed what a prosthetic group is. Okay, so it's also a cofactor. But your holoenzyme is your apoenzyme and your coenzyme. Okay, so this would be its definition. So the enzyme without its non-protein moiety is termed as your apoenzyme, and it is in your inactive form. Okay. Your holoenzyme is an active enzyme with its non-protein component, and that is your cofactors. So your apoenzyme is your, again, apoenzyme plus your coenzyme. Okay. So again, this explains your prosthetic group. Okay. So we go back to competitive inhibition. Okay. So this one kind of increases difficulty. Okay? Hindi naman pwedeng always recall questions. So this one, you have to be familiar with common reactions, and you know if it's a competitive inhibitor or non-competitive inhibitor. How will you remember this? How in the world will I memorize all these reactions and know what reactions would be competitive and non-competitive? But usually, what do you remember? What is irreversible? What reaction? And usually this would be the reactions that you should remember in biochemistry.
Hexokinase.
What's the
By C6P.
characteristic of your hexokinase? O yun muna tanggalin natin. Hexokinase glucose 6-phosphate.
Hexokinase nag-a-add po ng phosphate.
You know that it is part of your glycolysis. And what is it? How would you describe your hexokinase by glucose 6-phosphate?
Pag plenty na po yung G6P, hindi na siya phosphorylate pa.
Okay. And what would you call that inhibition?
Negative feedback po.
Negative feedback. Okay. What else could you remove? Ayun na, pag nakita na yan, manghuhula na lang. Well, again,
Sorry po daw.
If you see this and you cannot remember, well, I hope your educated guess would be correct. But sometimes that educated guess would depend on also still your stock knowledge. Okay? So, may paghuhugutan naman kayo ng sagot. So, even if you say that I'm not sure of the answer, more or less, you know what would be the answer.
Dok, isa pa pong try.
Sige.
Yung cytochrome oxidase po ba by cyanide?
Hmm. Is that an answer or you are removing it?
Yung answer na po.
Your answer. Any other guess? Why is it not A? Ano ba kung ano? Ano na eh? Why is it not A? Or why is it not B? B is also non-competitive. Actually, the answer is letter A. So, you have to touch on your common reactions and review. Kahit even if your reactions, you do not memorize it all. I know your whole cycle, when you talk of it, you will take an exam of it. It's quite taxing to memorize your glycolysis, gluconeogenesis, citric acid cycle, your Krebs cycle, but at least memorize those four or be familiar with those four. Okay? And the steps that would have rate limiting steps. Okay? Where there is a block, when there would be reactions that would be irreversible once it proceeds. So those are the important reactions in our body that you have to take note of. I know there are quite a lot, but at least in your main cycles, such as your um, glucogenesis, glycogenesis, Krebs cycle, citric acid, for your non-carbohydrate, your pentose phosphate pathway, and the like. Okay? So remember your reversible competitive inhibition. So you remember the concepts of your Vmax. What do you have to remember with these concepts would be your Vmax and your
KM ba, Doc?
Km. Okay. Your Vmax and your Km, remember with your reversible competitive inhibition, what happens? When Vmax is increased, what happens to your Km? Remember our diagrams for that? Nakalimutan na. As to your non-competitive, your Vmax and Km, what is its relationship?
Half po ng Vmax yung Km. The amount of substrate to reach half po ng Vmax. Yung
But it's
Km.
very much affected by your Vmax. Is it your competitive or your non-competitive inhibitor? Medyo nakalimutan. If you forget which is which is greatly affected by your Vmax, your substrate. Is it competitive or your non-competitive? Which of these reactions, okay, that if you increase your substrate, it could reverse, ayan na nga sinabi, reversible competitive. It could overcome your binding of another substrate to your active site. it is your competitive inhibition. You remember my analogy with this, that if in a relationship, both of you would want the same girl or boy, your competitor is already binded, but you are the one who wants to go in that position, you exert more effort. Okay. If it's not competitive, what happens? So when it is already binded, even if you increase your substrate, it would not change, it would not accept you anymore because like something else, and it's not the same configuration as yours. So even if you do a lot of effort, it would be futile. So even if you increase Vmax, your Km will not change. Okay, we will go back to that later and we will shift to that power slide. I think you need more review with that. Okay. So while bound to active site substrate, the substrate is converted into the product of reaction, a complex, another substrate of high energy. So bound to the active site, what is it? It's the enzyme substrate complex. So is it a product? Not yet. It's a complex, yes. Another substrate of high energy. So it is a complex. Okay. So remember your enzyme substrate complex. So your enzyme and your substrate and then forms into your enzyme substrate and your enzyme product. And then eventually your products will be produced. All right. Did your enzyme get used up?
No po.
Okay. What theory is that or what concept is that if you remember your biochemistry 1? Ayaw na ayaw. Hate nyo. Lagi. We will go back to that because most likely you dump it outside your stock knowledge. Okay. Enzymes do not work in high temperature because sana ganito ulit ang questions, no? What's the answer?
They get denatured.
They get denatured.
The enzymes get denatured because enzymes are protein. Okay. So review. So this would be asking you the concept of what affects your enzyme activities. Okay. So of course, there are 3 factors. You have your environment, you have your cofactors and coenzymes, and you have your enzyme inhibitors. Okay. This question is asking about your environmental conditions. And if you do remember, aside from temperature, what affects your environmental conditions?
pH po ba daw?
pH is 1, temperature is 1. What's the other? Let us repeat. Okay. So extreme temperatures are the most dangerous. You know that at more than 40 degrees, your protein gets denatured. But of course, approaching that, more heat excites your cells, thereby it gets more excited. That thereby your rate of reaction increases faster, but not exceeding extreme temperatures. Okay, because it may denature the enzyme. Okay. The second is pH, and most would like your 6 to 8 pH which is near neutral, except for some who would prefer your acidic environments, which would be, of course, your stomach enzymes such as your pepsin. Okay. The third would be your substrate concentration. Of course, more substrate would mean faster reaction, but it has also a limit. And why is there a limit with substrate concentration, as you have learned in your basic chemistry?
Because Doc, pagka yung ano po, yung sites kung saan pupunta yung substrate, pag napuno po yun, parang magmamax na siya.
Okay, you have your Vmax as your receptors get filled up. Of course, even adding more substrate, so that is the concept of supersaturation. Um, your reaction would not go any faster. Well, in anything, in economy, that's supersaturation, that's law of diminishing returns. In chemistry, that's supersaturation and your Vmax. Okay. So this would be your optimum temperature. Anything more than your 40, your protein gets denatured, thereby affecting your rate. Okay. This is your pH. Most would like your 6 to 8, except for your stomach enzyme, particularly pepsin, which would prefer your acidic conditions. Your trypsin would lean towards your basic pH. Okay? And of course, this is your substrate concentration and reaction rate relationship. Okay? So as your substrate increase, it gets faster, but it has a maximum velocity and it stops increasing the rate of reaction. Okay? Okay. Other factors would be your cofactors and coenzymes. So this would be your inorganic substances and vitamins that we have mentioned in our previous slide. And they are needed for proper enzymatic activity. Again, your supplements are fond of this. They give you coenzyme Q, they give you vitamins, etc., etc. And this is the reason why it is given, because this increases enzymatic activity, and therefore, if for you know that your certain diseases would have enzyme deficiencies, and these cofactors will help a lot. Okay. So iron, they must be present for hemoglobin to pick up oxygen. Okay. So one of the more basic cofactors would be iron, and how it affects your hemoglobin as it circulates around the body. And more often, you look at your hemoglobin, and sometimes you fail to see that your iron may be at your normal levels, but the problem is your ferritin, and your total iron binding capacity would be the problem. And you just give iron blindly. So it brings more harm clinically, because you have your excess iron, which is also bad. So you have to investigate further and not just accept low iron. You have to give iron left and right. Sometimes giving blood transfusion is not always the answer. Okay? Okay. That would be for the questions. I think I will have to review you with your enzyme kinetics a bit. Let, let me just share that slide, that PowerPoint. Sana yun na lang, no? Enzyme kinetics exam. Okay. Okay, so this is your favorite. Okay, so this is the one I was talking about earlier. And do you remember, again, your Michaelis-Menten equation has three concepts or three premises. Okay, this is the first premise. And this was shown in one of the questions asked earlier. What is the relationship of your substrate and your enzyme? What's the first statement of your Michaelis-Menten equation? Talagang nilagay sa baol, kinalimutan. Using that diagram, I know those premises are wordy, okay, but I want you to remember the concept. So what's the relationship between your S and your E? Your substrate and enzyme to make your Michaelis-Menten equation statements.
Mas marami pong available na substrate sa enzyme.
Your substrate is so much greater than your enzyme such that
Pwedeng ma-fully saturate yung enzymes.
fully saturated, such that your enzymes do not get used up. That the concentration of your enzyme is negligible to the equation when you derive your Michaelis-Menten equation. Okay. It's also assumed that your energy substrate does not change with time. So rate of synthesis is equal to rate of degradation. Of course, you know that this assumption cannot be true because there would be fall-offs, natural losses that occur with any reaction, especially reaction in the body, any organic reaction for that matter. And we use the initial reaction velocities. Okay, such that if you use your initial reaction velocities, your other side of the equation is very much decreased because your initial reaction velocity could be equated to 0, thereby simplifying the equation. But you know that this Michaelis-Menten equation mechanics makes things simplified because it follows your single substrate reactions. And you know that most reactions in our body are multi-substrate. Okay? So your Km. Okay, so we go back to your Km. Your Km, so that's your constant, is specific for a certain enzyme for a particular substrate, and it reflects the affinity of the enzyme for that substrate. Okay, so a large Km so means there's a low affinity of the enzyme for the substrate. Okay, if a small Km, it reflects a high affinity of enzyme for a certain substrate. And how does that how would this be put into play in your competitive and your non-competitive inhibition we'll look at later. Okay. So conclusions for your Michaelis-Menten, your rate of reaction should be directly proportional to your initial velocity and your Vmax. Okay, again, high concentration, low affinity, low concentration, high affinity, and low concentration would be first order, and your high concentration would be zero order. Because low affinity, so it would not be dependent on your substrate concentration. If you're low concentration, high affinity, it is proportional still to your substrate concentration. Okay? Okay, so this would be your inhibitors. Okay, so in competitive, okay, your Km is increased, your Vmax is the same. Okay, so for competitive, again, your Km is increased and your Vmax is the same. For non-competitive, your Km is the one who is unchanged and your Vmax is decreased. Because remember, in your non-competitive, even if you increase that, it would not change your reaction. Okay? And this is your negative feedback. Okay? I think that's it. Any questions for the review? I think I am just for 10 questions, if I remember correctly. So I don't know if it will be used for all. Usually, when it's a seminar exam and you have a lot of topics, per topic you get around 3 to 5, maximum 10 questions each. I think they asked 10 questions from me. So per topic, be prepared to answer 3 to 5 questions. So you have a lot of other topics to cover as well. So testmanship tips, know the must knows, okay? Know how to eliminate, the process of elimination, and if you're taking too much time on one item, you can skip. Although in your online exam, the dynamics get a little bit different. Okay? It's hard to skip questions and hard to go back to your previous ones that you don't get answered. Sometimes you end up not giving an answer at all to a question that you were not able to go back to. So for online, make an educated guess and don't second guess yourselves. You have learned this in your first year and more or less believe in your hunches. Okay? Unless you are very sure of the answer that you will change. Okay? Any questions? I will leave you with the afternoon to review for your exam tomorrow. It will be through Zoom. Link will be provided. I will provide the link for you tomorrow. Okay? Any questions?
None po, Doc.
Okay. If there's none, you may leave this call. See you tomorrow. Have
Thank
a
you
good
po, Doc.
Thank you po, Doc.
Thank you, Doc.
Thank you, Doc.
Make sure your internet connection's okay. Sometimes like it or not, your internet speed matters. So get a good signal so that you could concentrate on the important things. So remove all things that you can control. So make sure that you have a good space, you have good quiet space to answer, you have good connection so that you could concentrate on the exam. I know there are unforeseen instances during an online exam. Sometimes you submit but it does not load, etc., etc. But we try to prevent anything else so that it will be uneventful. All right? But please do inform us if there's any problem we do consider. Okay?
Okay po, Doc.
All right.
Thanks, Doc.
Thank you,